Professor Dash, we'll request you to see whether you can see it from your end.
Yes, ma'am. Sir, is it visible? Hello?
Abdul
Sir?
Ghani, if you could please turn off your video, that will be great. Thank you. Abdul Ghani.
Yes, sir. Turning off. Thank you.
Thank you.
Hello.
Hello?
Can you hear me?
Yes, sir. But it's very static. Your voice. Hello?
Yeah, I said there's a power cut in our area. Hello.
Yes, sir. Yes,
Hello.
sir. Yes, sir. You can be heard.
Can you hear me?
Yes,
yeah,
sir.
I said that there's a power cut. Come on. There's a power cut in our area. Our battery is also exhausted.
Yeah.
So basically, I'm waiting power to come. Can you start in 5-7 minutes? Let's see if the power comes in.
Yes. Okay, sir.
Yeah.
Yes, sir.
I think there is some time delay. Actually, that is what happened. Yeah.
Shinjini, why don't you
Yes,
start? Shinjini, why don't you start?
Yes. Just a second. Yes, ma'am. I'll start now.
Hello.
Yes, sir.
I think rather you start. Let me carry on as long as whatever the remaining battery can sustain.
Okay, sir. Okay, sir.
Someone who is chief arbiter.
Okay. So, good evening, everyone. We are sorry for the delay, a little bit of delay that we had. Welcome to the Progress and Prospects in Biology webinar series. Today's lecture is being delivered by Professor Gobardhan Das of the Jawaharlal Nehru Institute, New Delhi, on the topic of BCG, a century-old vaccine against COVID, science and sense. So now, before we start, there are a few housekeeping rules that I would like to remind everyone. Please make sure to keep your microphones muted and your videos off during the lecture, and do not share your screens at any time. Otherwise, we shall be forced to remove you from the meeting and not readmit you. I request to everyone to keep the presentation window pinned to your screen so that we can have an uninterrupted session. Please do not use the chat box to introduce yourselves. Just post questions there for the speaker, which we shall address at the end of the meeting. At the end of the lecture. And the link for the feedback form shall be given at the end of the meet, and the link will be open till 8:30 p.m. only, and it will not be shared to you via email. So now I would request our mentor and convener, Professor Rena Ray Banerjee, to welcome our speaker. Ma'am?
Yeah, thank you, Shinjini. I'm having some issues with my device also. I
Okay.
hope Mr. Dash is with us. So, and everything will go as planned. We are having too many glitches. Anyway, um, maybe they are good signs. So, a very warm welcome to all the participants in tonight's webinar by Professor Gobardhan Dash. I have the privilege to introduce the speaker this evening, and he will be speaking about something that I think is the number one question on all our minds. When is the vaccine for COVID going to appear on the horizon, and when we can go back to our normal life? So I think today's talk promises to discuss some really pertinent issues. So a warm welcome, Professor Dash. I will introduce you now. He is our today's speaker is the professor in Special Center for Molecular Medicine, Jawaharlal Nehru University, New Delhi, and the adjunct professor in Houston Methodist Hospital, Houston, USA, and Shuchao University, Suzhou, China. I hope I have pronounced that right. He was the visiting professor in Shuchao University, China, University of Technology, Australia, University of KwaZulu-Natal, Durban, South Africa, Johns Hopkins University, Baltimore, USA, University of Turku, Finland. And I hope we have viewers from there tonight. He's the professor in the University of KwaZulu-Natal, Durban, South Africa. Is the senior scientist ICGEP New Delhi and Aventis Pharmaceuticals New Jersey, USA. He is the recipient of a number of awards. Of them, he is a fellow of the Indian Academy of Science 2018. He's the fellow of the National Academy of Science 11, Academy of Sciences and Technology. Trust DB Senior Fellowship and 9, and Ramalinga Swami in 2000. Also the National Africa 20 Awards. He is associated with molecular immunology from Gubo Research Council and American Association of Microbiologists. He has trained 17 PhD students and 22 postdoc scientists under his guidance and has published 70 research papers, 3 book chapters till date. He is also the associate diseases and reviewer of many reputed journals. Okay, that's a substantial introduction. A very warm welcome to Professor Dash, and we all are waiting to listen to you. I know we have a bit of a technical glitch here and there. I know you have a power outage in your locality, but despite that, we all are looking forward to what promises to be quite a stupendous talk today. So with that, it is over to you, Professor Dash.
Yeah. Thank you. Am I audible?
Yes, sir.
Hello?
Yes, sir.
Am
You're
I
audible.
audible?
Yes, you
Yes,
are.
sir.
Yes, you are.
So, thanks for the generous introduction. And I sincerely thank all the organizers for inviting me today. And it's really a big opportunity to inform some of this laboratory focus is tuberculosis. Recently, because of pandemic, also I am giving hands to on COVID research as well. So recently, some of the things what we have done and what we believe we'll be talking about today. So as you know, this is COVID is a very brand new disease, and it's that's what it's called novel coronavirus.
Sir, are you?
I think our speaker is having some issues at his end.
We are sorry for this delay, but Professor Dash is apparently appears to have some technical problems today. We'll be starting shortly again, so please be patient and bear with us.
Hello.
Yes, sir. Hello? Sir, am I audible? Sir, I think you'll need to unmute yourself.
Can you hear me?
Yes, sir, I can hear you now.
Can you go to the next slide?
Yes, sir. Sir, maybe you can keep your video off so that we can preserve a bit of the bandwidth, and maybe it'll solve some of your problems.
My video is not on. My video is off.
Okay, sir. Okay. Yes, sir.
So on and off, as we know, we are right now at this moment, we are in the middle of pandemic, and pandemic is caused by the novel coronavirus we call SARS-CoV-2, and but on and off we are getting several in various kinds of diseases, and what is basically what we believe is because of you know this is because of human intervention with the environment, and right at this moment almost all the countries, 205 countries so far affected because of this coronavirus. And so that's what I put this slide. There are lots of lockdowns going on throughout the world. And can you click next?
Yes.
So basically, that's what I put this slide and say.
So your voice is breaking, actually. Hello? 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 Hello, sir? Sorry about that. I think sir has left again and he's having some trouble with his network, I guess. Just hold on.
Hello.
Yes, sir.
Yeah, is often is being disconnected. I'm
Yes.
getting hard time to reconnect actually, and somehow I'm asking you to next slide. You somehow you don't. It looks So can you go to the next slide?
Yes, sir. So we couldn't hear you actually.
Yeah. So that is what the see in this slide. What I'm trying to show you, the majority of the global countries throughout the world is now affected. But if you look at carefully, the intensity of the color is very different. It greatly varies. Why this is? That means that basically there is a degree of susceptibility or degree of occurrence of COVID-19 in greatly varies countries to countries. So we tried. Our first question was why these various countries are differing in their susceptibility to COVID-19 infection. So consistently, to break this long story short, consistently what we found, one variable that now has perfect correlation with the severity or the incidence of this COVID-19 infection. If you look at, can you hear me?
Yes, sir.
Yeah. If you look at this, I have listed some of the countries in black letters in upper portion, as you see, USA, Spain, Italy, and France. They are in black letters, and the middle column in the black letters is respecting numbers at the per million incidences per million population. In USA, more than 17,000, Spain more than 8,000, Italy more than 4,000, so on and so forth. And the extreme right, this is once again the in a black letters. This is the mortality per million population: 536, 616, and 586 and 487, respectively, to the countries USA, Spain, Italy, and France, respectively. On the other hand, if you look at the lower portion, in blue letters, countries like Thailand, Japan, India, Greece, they have the middle column in the blue letter is C49, 463, 2103, and these are the numbers per million population and incidence per million population. And the extreme right, this is 0.8, 09, 40, and so on and so forth. These are the numbers of mortality per million population in these countries, Thailand, Japan, India, respectively. And so why there is the difference? Only difference what we found, the black letters countries, they have the they do not have BCG immunization program. On the other hand, these countries, the Thailand, Japan, India, Greece, these are the countries, they have universal BCG immunization program. Next, please. So now that I'm coming to what are the symptoms of COVID-19 infection and what the initial symptoms. The person who is suffering from the COVID-19 infection or the symptomatic infection, the person is having fever, chest pain, chills, rapid heartbeat, breathing difficulties, pneumonia, and kidney failure. When it spreads, this spreads to the droplet through sneeze or cough or speaking. That is taken up by the naive person. And the initial symptoms of the disease is headache, sore throat, cough, and shortness of breath. Next slide, please. Next slide, please. So where this disease come from? Where this virus come from? So as we all know, this has come from China, the Hubei province from Wuhan city, from a fish market. So if you look at from where this animal planet, where it is coming from, it is universally believed that it is coming from the bat coronavirus, which is SARS-CoV, also known as RATG13. It has 96% homology with the current coronavirus, which is causing the pandemic. So these are the similarities. What is the difference between bat coronavirus and this SARS-CoV-19 is as we know, this virus enters to the human cell, human epithelial cells. There is a receptor we call ACE2, angiotensin receptor 2. Basically, it binds with the spike protein. Within that spike protein, there is a domain we call it receptor binding domain. In this domain, there are 6 residues plays important role for anchoring. Among these 6 residues, 5 of them are mutated. So basically, there is a big difference. And in addition, in human coronavirus SARS-CoV-2, in this spike protein, in between there's S1 and S2 domain, there is a 12 nucleotide insertion. So these are the 2 differences. So now from there, it was people thought, can it be directly come from the bat? The popular theory is no. So basically from bat, it's jumped to the another animal, which we call it pangolin. Pangolin is a rat eater, which is the Chinese delicacy. And if you look at the coronavirus from the pangolin, which has a little less homology, but all the 6 residues in this RBD domain as very similar to the human coronavirus. And some other people think this is the man-made coronavirus, and that's because of it can be generated from the bat coronavirus and so on and so forth. So I'm not really trying to enter in that controversy. Can you go to the next slide, please? So why we are getting this kind of diseases? What we believe is what is happening on the earth is not really correct.
Hello, sir. Hello. Sir, you cannot be heard. Professor Dash, can you hear me? Hello?
Can you hear me now?
Yes, sir.
Yes. Can you put back the slide? I'm just straight from the phone. I'm trying.
Okay. So which slide this disbalance on nature? This slide?
Yeah. Can you? Yes.
Is it visible now, sir?
No, I don't see anything.
Okay, I'll just share it once more. So is it visible now?
Shinjini is presenting, but
Yes,
slide is slide is not visible.
Oh.
I don't see
Okay.
slide.
I'll just
Yesterday I requested to do the Zoom. Rather, this is Google never works. I mean, every time I have hard time with Google, but so that's what I prefer always in Zoom.
If Zoom was possible, we would have definitely done Zoom. Unfortunately, we don't have a subscription, which I have to.
is free.
Exactly.
So is it still not visible?
No, every whole screen is dark.
So now I've shared it again.
No, it's not. You see, I can see myself.
Uh,
Oh.
Professor Dash, can it be possible for you to
So can you pin my screen to your screen?
I'm trying to do that now.
the last thing, a 3-year-old Syrian? So would you be able to do that?
I don't have slide in front of me. No. Oh, everything is gone now.
So you can change the layout to, I think, speaker mode, the presentation screen to
Let me.
view all the parts.
Yeah. How do I?
The 3 dots at the bottom of the screen, at the
Yeah.
right side bottom, you can click on that. There is a change layout to view all participants. You should
Switch
be able to
camera, turn on your caption, present screen, report problem, report abuse, help, cancel. There is.
the
Yeah, so I can see my screen, your slide now. It's this slide says this is the right now. That's what we are doing is because of what we believe, because of what we are doing on this art is not correct, and wherever we get some space, we try to do some kind of imbalancing to this mother earth, and that's what I put this slide. And as you see, 3 years old Syrian boy, he also knows what's happening on this earth, and that's what before he was dying, and the last word line he uttered, I'm gonna tell God everything. So, but basically, that's what we are trying to. All we are trying to say, what's happening on this earth is not correct. So, next slide, please. So now let us look at the virus. So, what is this virus? As I'm not going to go in detail, but in brief, this is a RNA virus. That means its nucleic acid is ribonucleic acid, and it has like other viruses, it has a code protein, and also one of the most important protein is called, we call it spike protein. This spike protein plays an important role, or this is works as a ligand for the receptor on the human cell, as a human epithelial cell. In this spike protein, there's a portion in S1 domain, we call it RBD, receptor binding domain, that binds with the ACE2 receptor expressed on the epithelial cells. As in the right side, as you see, from there, what we see is ACE2 binding with the ACE2 that has been endocytosed, and after endocytosis, this RNA comes out to the cytosol, and because this virus has its own RNA polymerase, dependent RNA polymerase, it can utilize the cellular machineries and to generate much number of RNAs as well as the proteins that's finally been assembled and then come out and ready for the ready for infecting the next set of cells. Next slide, please. So what are the immune responses and how it generates? In left side, as you see, when it is taken up or the endocytose within the cell because of the virus RNA that activates the various innate immune components that leads to the activation of various transcription factor, and therefore it produces several inflammatory cytokine and inflammatory mediators, which has been now come out of the cell, and then that leads to the activation of the various cell types. That's including macrophages, NK cells, various T cell subsets, etc. And as a resultant, we get activation of both innate and adaptive immune components. And finally, at the end of the day, we get two kinds of cytokine production or the immunity. If this cytokine production leads to the IL-15, type 1 interferon gamma, IL-12, IL-21, etc., that leads to the virus clearance. On the other hand, if we get IL-6, etc., IL-TNF alpha, IL-17, GM-CSF, that leads to the hyper inflammation, and that carried out and that finally caused the inflammation, and that is the cause the pathology. Can you go to the next slide, please? Okay, so what kind of inflammation? As you see in the left panel, you see in the lung, this person lung, as you see, there is several spots, and we can see inflammation in both the lobes of the lung. If you look at the right panel, I'm talking about panel A, where as you can see, the proteinaceous fibrin exudes, as you can see very well, which is grayish type. And then B, as you see, the thickening of fibers, fibrosis, and then in C, as you see, fibroblastic foci. That means where the inflammation already started. And finally, the D, as you see, the huge infiltration of polymorphic polynuclear cells. And finally, what happens is the whole lung lobule is filled with inflammatory cells and also filled with inflammatory milieu. Next slide, please. So what are the intervention right at this moment we have? As we all know, this virus is a novel virus, only a few months old. And so, as we all know, for the development of a drug or a vaccine, it takes several years. So therefore, at this moment, neither we have a specific vaccine, nor we have a specific treatment. So all we are currently depending on the repurposing drugs and repurposing vaccine. The repurposing two of the drugs I'm listing here. Other than these two, there are several others also being tried, are being right now in clinical trial. Two of them are making noise, lot of noise, the remdesivir and hydroxychloroquine. Remdesivir is the one which is antiviral drug, actually developed for Ebola virus, but it really didn't work for Ebola virus. It been tried for the SARS-CoV-2 in some of the places. For example, in China, they tried, but it really didn't work. But in some of the places in US, there are some efficacies it has been reported. So a bigger clinical trial is going on at this moment. Because hydroxychloroquine, on and off, we get good news and bad news oftentimes. And as is many of the times, still people believe this is hydroxychloroquine has some kind of efficacy, and the big trial is going on right at this moment. And next, I'm coming to the repurposing vaccine. As we know, for the development of a vaccine, it takes several years, and we indeed there are several vaccine is now being in under development development. In the meantime, there are several countries, including US, UK, Germany, and Australia. They are all trying for the, including India, trying for the BCG immunization, reimmunization. And I'll come to that point in a couple of minutes. Next slide, please. So this is in this slide says the current status of vaccine. This is a little old slide, but right at this moment, more than 200 vaccines are under are being developed, and 8 of them are under clinical phase 3 clinical trial. And if you look at carefully, the all sorts of strategy being tried. It's like DNA, RNA, inactivated virus, live attenuated virus, non-replicating viral vector, protein subunit, and replicating virus and virus-like particles. These are all the strategies is being taken throughout the world for the developing a vaccine against this novel coronavirus. Next slide, please. Next slide, please. Okay. So now the question is, what kind of vaccine we need? And to understand the kind of vaccine we need to understand the immunology, what kind of immune response for the protection against virus we need. We need both innate immune responses as well as adaptive immune responses. From innate immune responses, we need one of the biggest lion's share of the innate immune response against virus is provided by the type 1 interferon alpha, and mostly interferon alpha and beta. As we all know, various virus for viral infection, type 1 interferon is given, which is one of the treatment for the viral infection. Type 2 interferon plays also important role. In addition, natural killer cells as well as macrophages play important roles for the activation of innate immune responses. Can you go to the next slide, please? What are these innate immune responses? What they do, as I mentioned, type 1 interferon. Type 1 interferon actually blocks several viral replications and inhibits the viral protein synthesis. Some directly or indirectly it degrades the viral RNA, and inhibition of expression as well as the assembly of virus particle. So, and next, natural killer cells. What natural killer cells does actually directly kills the virally infected cells. It somehow recognizes virally infected cells. I'm not going to go in detail. It can sense, natural killer cell can sense virally infected cells and immediately go and kill the target cells. Generally, upon recognition the target cells, they start producing perforin, which cause the pore on the target cells, and that injects this granazyme, which degrades the DNA in the target cells. At the end of the day, this is target cell is being killed. Next is macrophage, which plays an important role for elimination of the virus directly or indirectly. It phagocytoses the infected cells, as well as it can engulf and directly kill this virus. And also it activates the innate immune responses that leads to the production of various effector molecules and the cytokines such as TNF alpha, nitric oxide, and interferons. So next slide, please. In adaptive immunity, we need 3 things: the antiviral antibodies, cytotoxic T lymphocytes, as well as T helper cells. As you see from the left, what we are showing, trying to show you the antibody, where the antibody blocks the binding with its receptor. So therefore, rather, this antibody binding facilitates the phagocytosis. Upon phagocytosis, the macrophage or the phagocytes, they engulf and then finally degrades this virus particle, and that leads to the activation of phagocytes, and then finally they produce various cytokines and effector molecules such as various cytokines, nitric oxide, etc. In the right panel, as you see, this is cytotoxic T cells. Cytotoxic T cells, they recognize the virally infected cells. Upon infection with the virus, in the cytosol, the protein is synthesized de novo synthesis of protein that is available for the proteasome complex, and that is generate the small peptides that finally have the access with the MSC class 1 antigen presentation pathway. That is viral protein or viral peptide is being presented with the context of MSC class 1 molecule that has been recognized by the cytotoxic T cells, which are often CD8 positive T cells. And upon recognition with virally infected cells, CD8 T cells start producing the secreting perforin, which makes the hole on the target cells, and then secretes the granozyme, which degrades the DNA. At the end, the target cell is being killed. Next. As you see, this is, I also listed the T helper cells. T helper cells is required for the activation of the B cells. And in fact, the T helper cell, which helps the B cells by supplying several ligand receptor interaction, as well as the cytokine. For example, IL-4, IL-5, IL-13 plays important role, not only the growth factor of B cell, but also required for the class switching. So at the end of the day, you need the T cell help to B cell for the production of affinity matured antibodies, and that antibodies finally can neutralize the virus particle. Next. So we are talking about BCG vaccine. What is this BCG vaccine? BCG vaccine is invented in 1921, which is almost century-year old. So why I'm talking about a century-old? That's because it has been tried for that long, almost 100 years, and it is given at the birth, at the one day old children. So that means this vaccine is very, very safe, and it has been efficacious for disseminated TB and meningitis in children. Unfortunately, it loses its efficacy, and it really doesn't protect from adult pulmonary TB. So during this 100 years, we have learned that it has been developed for TB, but it also have the off-target effects. It protects from the leprosy. It can be used for the Burell's ulcer, and it is given for the bladder cancer as well as in colorectal cancer. Several other other also being tried. For example, for colon cancer, sepsis, and type 1 diabetes, it is being now under trial. One of the most important thing I would like to mention here, BCG has been given to rhesus monkeys in
Hello? Sir has some trouble with his network, so I'm sure he'll be back right now. Hello, sir. Sir, you'll have to unmute yourself.
Yeah.
Yes, sir.
Yeah. Can you slide, please?
Yes, sir.
Where's the slide gone?
Okay. So I think you'll have to change your layout once more, maybe.
Where did I do that?
3 dots at the bottom right side of the screen.
Yes. Right? Hello?
Is it visible? Yes, sir. Is it visible now?
Yes, it's visible to me.
Yeah.
Okay. So one thing I would like to mention here that the rhesus monkeys, which has been immunized the BCG. Later in the life, these monkeys have been protected against several respiratory infections. So not only that, there are currently BCG is being in clinical trial for COVID-19 in several countries that includes UK, Australia, Germany, right now in India as well. Next slide, please. Yeah. So here is some when this coronavirus or COVID-19 is being spread from China throughout the world. In early in the mid March, we are trying to understand why the various countries are affected in various degrees. So one of the things we consistently found it has some kind of inverse relation with BCG immunization. Then we segregated these countries, those who we have taken the countries, those who have more than 1,000 incidences, and then segregated the countries in three parts, either those countries who never have BCG vaccination, and those who have universal BCG vaccination program. And in the middle, as you see, there are some of the countries, those who had this BCG vaccination program and then discontinued. So we started looking at the incidences per million cases, as you see in the y-axis, the cases per million in log scale. In the countries, those who never had the BCG vaccination program, their incidences is much higher with compared to the countries, those who have the BCG, universal BCG vaccination program. Exactly in the middle, which those countries who actually discontinued BCG vaccination. Next slide, please. So this is the now I'm coming to the death per million. As you see, once again, the countries, those who had no BCG vaccination, they have much higher incidences than than those countries had the universal vaccination program of BCG. And in between, we see those countries who discontinued the vaccination program. Next slide, please. We all know the BCG, the COVID-19 susceptibility to mostly the elderly people. Actually, the children are not much affected. So therefore, what we wanted to look at was the age distribution and then compare incidences per million population in three types of countries, either no vaccination and discontinued vaccination, and those who got the universal vaccination. As you see in the upper side, which is showing the age group of 15 to 44, and the lower panel, which is 45 to 64. And everywhere, what we see is the incidences per million population across the age where there is no vaccination as much higher than those countries who had the universal vaccination. Next slide, please. In next slide, once again, this slide, once again, it says the age group is 65 to 79 and more than 80 years old. As you see here also, what we see incidences per million population where there's no vaccination countries has much higher incidences with compared to the countries who had the universal vaccination program in all the age groups. Next slide, please. Next slide. Yeah. In this slide, as you see, we have taken the mean, all these countries either having no either universal vaccination or the drop vaccination or the no vaccination. In the lower blue line, as you see the countries, those who got the universal vaccination, their incidences much less than the countries who are in the middle, as you see, which is the green line, those who dropped the vaccination program, and also those countries which is shown in those countries who never got the BCG vaccination, which is depicted in red line. So basically from here, what we conclude, basically at least the incidences per million population is less in the countries who has the universal BCG vaccination program. Next slide, please. Next slide, what we are comparing, like in incidences, we are comparing the death per million population. Once again, 3 types of countries, either having no vaccination or the universal vaccination or the discontinuous vaccination. From here, as you see in the upper panel, you skipped one slide anyway, so I really don't need. So all these age groups from 15 to 44, 45 to 64, and 65 to 79, and more than 80, all the age groups, wherever we see the mortality by the coronavirus, COVID-19, death per million population in those countries who had no vaccination of BCG is much higher than those who are having universal vaccination countries, as shown in here. So next. Yeah, here. In this slide, what you see, the blue line is having the mortality per million population in countries where the universal BCG vaccination program. In the middle, you see there is a green line where the countries who dropped the vaccination, BCG vaccination program, and red line where you see there's the countries who never had BCG vaccination program. Next slide. Next slide. From where what we have learned, that basically BCG vaccination has some kind of inverse relationship with the COVID-19 incidence as well as the mortality. Now the question is, where these various countries been immunized with the BCG, and all these countries are doing the equally. We don't think so, as you have seen from the scattered plot. So all the countries are not doing equally well. So from what we feel, probably the various strains of BCG being used during these 100 years of subcultures, there are various strains are being generated, and because of use of various strains, their protective immune response is also varied. So as you see from the left panel is incidences per million, and the right panel is death per million population. Some of the BCG strains, such as mixed strain, Japan as well as the pasture, they are protecting very well. On the other hand, probably BCG Russia, BCG Denmark are not really doing that well. Next slide, please. From here, what we have learned, both incidences as well as the mortality it has some kind of inverse relation with death as well as incidence as well as the mortality due to COVID-2 or COVID-19 as inversely proportional with the BCG vaccination program. Now the question is, how long the BCG immunity remains? As we know, the BCG is given in the early childhood, and that's the immunity fades over a period of time, and it really cannot protect the adult pulmonary TB. So how this is we are being protected? What we believe this is, as you know, this is although the immunity against the protective immunity against the TB may not last long, but some other immune responses, it remains. For example, the leprosy. Now the question is, do we need boost? Indeed, what we need boost? What we believe we need boost, that's because in some of the countries like India, Brazil, those who have relatively less hygiene index countries, we are often being exposed to the various organisms, and how often we believe that also exposed to the environmental mycobacteria. So therefore, what we believe is nothing but is actually almost like a booster dose or the revaccination. So we really cannot depend on the revaccination. So therefore, what we believe, what we propose to revaccinate once again with the BCG. So now the question is, BCG is a non-specific immunity. Well, that's because, as you know, BCG is a bacteria. How it can protect against viral infection. There are several papers that are coming out, and it says indeed BCG can protect against COVID-19 by something called trained immunity, or also called this innate memory responses. So do we need really specific vaccine? Indeed, we need a specific vaccine. That's because we really cannot depend on the something non-specific. So next slide, please. Next slide, please. Okay. So I really don't want to go in detail, but all I'm trying to say, early in the mid-March, I wrote to the several government agencies as well as ICMR DG and Science Advisor to PM. We should start immediately immunizing or revaccination with the BCG or the MIP. MIP is Mycobacterium indicus prani, which is our indigenous strain, which is a little better than BCG in antigenically, because it produces more in type 1 interferons and produces activates a little more better innate immune responses. So that has not been hard, but after a couple of months, we saw various countries started immunizing BCG, revaccinating with BCG, and recently India also started the vaccination with various strains of BCG. Next slide, please. Next slide, please. Okay, so BCG immunity is non-specific, and regular BCG doesn't induce cytotoxic T cells. So right now, what I would like to talk about, what we are doing in my laboratory, we are trying to make it this immunity some kind of specific. How we are trying to do it? We are using BCG as a kind of an adjuvant. As we all know, we have to. There is a relation between BCG immunization with COVID-19 susceptibility. That means it is giving some kind of non-specific immunity. On top of non-specific immunity, we want to make it some kind. It is specific. So specific both for the cytotoxic T cell as well as the T helper cells. So how do we are going to do it? So next slide, please. So okay. So what we are right now, what we are trying to do is we are taking the COVID-19 protein, especially the spike protein, not the whole spike protein, actually the RBD, receptor binding domain. Recent literature suggests that only antibodies can neutralize this coronavirus is the antibody against this RBD domain. So what we are trying to do is we are taking out the RBD domain from the spike protein, and also for the cytotoxic T cells, only cytotoxic T cell can give the host protection is the cytotoxic T cells against the nuclear capsid protein. So what we have done is we have generated the recombinant of RBD domain as well as the spike and RBD domain as well as nuclear capsid protein. And then what we have done is we are expressing on BCG. So basically, as you see in the right panel, is BCG looks like inner portion looks like BCG and outer portion looks like a kind of a coronavirus protein expression. Next slide, please. Here, as you see, our vaccine will look like this. What is the advantage? Because of BCG base, which is a pretty good adjuvant, it can induce the type 1 interferons, also can activate innate immune components, and because of the presence of the proteins of the the RBD domain as of the nuclear capsid protein, we are going to treat the T helper cell as well as cytotoxic T cells against these proteins, and these T cells are going to help the B cells. So therefore, we are going to get the antibody response, and BCG is very good adjuvant, so it can give a memory response. So therefore, what we believe is at the end of the day, what it translates, the COVID-19 specific vaccine, which has both cytotoxic T cells as well as antibody responses, and because of the long-term memory, basically we can give it one dose, or at the most we have to give the a booster dose, and that can last for lifelong. Next slide, please. And this is where I would like to end, but at the last but not least, I have to acknowledge the people in the left upper corner. The Orna Bhattacharya is my collaborator, and the right upper corner is Vaibhav, is the student who did the all competition analysis, and the one left panel is Saurabh, is the new faculty. He has also done the statistical analysis, and this is my small lab. Right at this moment, we are working on both TB as well as COVID-19 program. I am funded by DBT and DST. So I would like to stop here. I'll be happy to answer any question you may have. I'm extremely sorry that it's not really went well. That's because of several reasons. Is all I can easily blame this network, and I'm really sorry, but I'll be happy to take any questions you may have.
Thank you, sir. We can't do anything about the technical problems.
Yeah.
It was a very enlightening talk in the light of the current scenario. So we have some questions here. So Shomajit Biswas has asked, there are different strains of SARS-CoV-2 virus all over the world. Do you think that one type of vaccine will be effective for all strains?
Well, this is a very good question, actually. Let me tell you, it's not the several strains, it's rather several clade. Anyway, so that's not the question. And, it is continuously mutating, that is true. And our strategy is little different, as although I did not mention, try to finish it quickly. And our strategy, what we are doing is we are taking several epitopes, T-cell epitopes, as well as the antibody B-cell epitopes. So we are stitching all these epitopes together. As we all know, the evolution is a slow and steady process, one mutation at a time. It's not in one day, it cannot mutate all of the epitopes. So therefore, even if the one epitope fails, the several other epitopes are going to protect. That is our strategy. So what we are trying to generate a vaccine strain, which is BCG is the base, and on top of that, we are taking the several epitopes from the several proteins, and then for T cells as well as several epitopes, B cell epitopes from the several protein. So we are stitching together and then make a generating recombinant BCG. Yes.
Okay. The next question is from Aparna Ramalingam. In COVID-19 recovered patients, it has been observed that antibodies aren't long-lasting, but neutralizing bodies seem to be. So what might be the reason? And why doesn't memory B cells develop even after an infection in this case?
Yeah, so that is what one of the things, another good question. That is one of the things, as we know, this is memory B cell antibody response doesn't last long, at least in COVID infection. There are several papers are coming out. All it says, the person who got the disease once, he may get the disease again. So, and it could be because of several reasons. Number one, of course, it's not memory is not being generating. There's memory is not being generating because of several reasons, because of vector and adjuvant property. It could be several reasons. So now, being those who are trying to develop vaccine, we have to stabilize, or we have to get the antibody which is long-lasting, and also can the memory response can be long-lasting memory. That is what we have chosen, not the viral vector, but the BCG vector. As we all know, when we try to generate antibodies, we immunize the animal, we use CFA, complete Freud's adjuvant, which has the mycobacterial part, as we all know. So that is what we are trying to take it. So in that same reason, because of same reason, I argued in several places in the government agencies that those who are taking the viral vector strategy may not work as because the viral vector is not very good for generating a B cell memory response. Number two, those who are taking DNA and RNA strategy exactly suffers from the same, same problem. They will get response temporarily and it will fade away in no time. And so it's not a long-lasting vaccine. That's why we have taken this strategy.
Yes, sir. Dev Mallo Sengupta wants to know, it has been observed that some patients are IgG responders and some patients are IgG non-responders. According to clinicians, the IgG non-responders were found to get severe ARS. Do you think this differential host immune response has some genetic cause? And if yes, can you provide some insight into this?
Well, I mean, see, this is always the host has the host genetics has always play important role in mounting various immune responses. And in fact, some of the antibodies may not work in some patients. That's because of viral shedding, where the virus is hiding and how the virus is being shed. So that entirely depends on the host genetics.
Okay. Siddharth Nath wants to know, in some region, certain age of people are getting autoimmunity or herd immunity. So is there any chance for those or to all the people in that region any harm, and why so?
If I did not get the question clearly, but what I understood the herd immunity, basically all we are talking about the herd immunity. Some people say the herd immunity in 15, 20, 30% infection is not. Herd immunity, how do we get herd immunity? It's simply a game theory. Basically, the A virus, one is trying to hit basically a person, and there's what is it? Basically blocking the chain, right? Or the terminating the chain. In a population, it has been calculated almost like a 68 to 70% population is being exposed to a given virus or the generated protected immunity against the A virus. It may protect greatly for the infection of a community. So that is what. Still, it doesn't say foolproof. So still in Delhi, I live in Delhi, or 23.9% people are being infected. Still, it's not the kind of herd immunity we got. So basically, we have to have some kind of at least 70% people who's been exposed or been got immunity against a given virus. It has also been calculated to achieve the herd immunity. So an exposure of the more than 70% population, at least we are going to lose 1 to 2% of the population. So in a country like India, 130 crore. So basically, we are going to get 1.3 into 2.6 crore population we are going to lose. Can we really afford that? We really cannot afford that. So that is what, so those who are talking about the herd immunity is not really the option. We have to have some kind of discovery of a new therapy, discovery of a new vaccine, and also in the interim, we have to take all the precautions as given by the government.
Okay, so Gaurav Bhardwaj's question is, as MHC class 1 gene shows polymorphism throughout the population, can it be correlated with the severity of infection?
This is also basically those who mount cytotoxic T cells, and the cytotoxic T cells are protective. Those who do not mount the cytotoxic T cells, against the protective cytotoxic T cells, they are less protective. That one way I can answer, but definitely it depends on the human genetics and of course the degree of the protective immune response depends on the human genetics and the polymorphism of MSC class 1. Not only MSC class 1, but also MSC class 2.
Okay, so Tushar Bokshi wants to know, and is there any update on the AZD1222 SARS-CoV-2 virus vaccine? Sorry.
See, this is so far. I know there are 8 of them are in phase 3 clinical trial, and those who are in clinical trial are getting some of them are getting good result, but whether they are long lasting, I do not know, because as you somebody rightfully mentioned, the immune response or the antibody response fades away fairly quickly, and also there are papers coming out. Patients are coming back once again with the reinfection. So therefore, so we are getting good immune responses in some viral, vaccination. Whether they are long lasting, that we do not know.
Piali Chatterjee's question is, India is in the 3rd position worldwide in terms of COVID-19 cases per million. USA being the top, followed by Brazil. So, but both Brazil and India have BCG vaccines. So how do you explain that?
Okay, this is a question is little I have to correct. Is what she says is not the per million population. If you look at the per million population, still our India is much doing much better than many countries if you look at carefully. And see, in India, we could have done is much better because of our behavioral. There are 2-3 things. Number one, behavioral problem, because of we are overly democratic country. So that one of the problems. Second problem is we have India is a now almost is the house of diabetic patients. So huge population with having comorbidity like diabetes and heart disease. Those who have the diabetes and heart disease and other comorbid symptoms, they are not as protective as it should be. So that there is a difference. Yes.
Sir, should I take some more questions? Or?
You can take as many as you want.
Okay, sir. So Obranil Gongopadhyay wants to know that there has been news that people who develop COVID-19 are also susceptible to develop tuberculosis. Could this be associated with the BCG vaccine in any way? Like if COVID-19 develops in a non-vaccinated person, it doesn't make them susceptible to tuberculosis by default.
So is this the question that those who got the COVID, they're also getting the TB? Is that the question?
Yes, sir.
Is the question that those who are getting COVID, they're also getting TB?
Yeah, there is a susceptibility of the people who are getting COVID, that they are susceptible to developing TB.
I never heard of it. In fact, there is a paper, those who got the TB, but are not getting COVID infection.
Okay. That's interesting. Okay, another question is Deboshri Golder. Is there any cellular mechanism which alters the expression of the ACE2 receptor in human epithelial tissues in human, inhibiting the binding with the S1 active domain of the spike proteins?
So is there any inhibitor for the ACE2 protein? So
yes,
there are several development trying from the all sorts of inhibitors, but so far there is nothing. No one is found so specific, and anything is under development is not in my knowledge, and maybe confidential. Something I don't know.
Okay. Also, there is another question: is if at present, if a person hasn't been vaccinated with BCG but decides to get vaccinated now, will he have less susceptibility to COVID? And are there any contraindications for getting BCG vaccine?
Well, I mean, see, this is even if we are not vaccinated, we are always trying to. We are proposing, yes, we should get vaccination. At least BCG, as long as it's a very safe vaccine, it can be given 1 day old child, and it doesn't have any harm. So basically, all it can do is you can do at some kind of immune response. At the most, it doesn't do anything. So it's no harm to get a revaccination. On top of that, we are in India, country like India, where there is a less hygiene index, we are often exposed to the various organisms. That includes environmental mycobacteria. So giving another dose doesn't really harm.
Another question is from Deepak Kumar Poria. Could age group of the population be a confounding variable rather than a causation of the claimed protection effect of BCG vaccine?
Wow. Anybody can answer this question? I don't know this. It's all you have to guess. I don't know.
Okay, so the last question is from Shobhagya Nayak. Can the upcoming vaccines combat with the mutations that coronavirus is undergoing?
As I said, see, this is depending on what kind of vaccine we are talking about, what the strategy we are being taken. So, for example, our flu vaccine, one has to take every year. So, in the coronavirus vaccine, if we get one, depending on the what kind of immune response we get, and whether it can really cover various mutated version of this virus, that will entirely depend on what kind of vaccine we are giving. The one vaccine, once again, I would like to re-emphasize what we are doing is we are not taking the whole virus. Our adjuvant or the base is not a virus. The other, we are taking the BCG or mycobacteria. And on top of that, we are taking the various epitopes of B cells and various epitopes of T cells. As I clearly mentioned again, the mutation is a very slow and steady process. So in a day, it's not possible that all the epitopes are being mutated and you get the disease. That's very highly unlikely. So that's what our strategy, what we are taking is to keeping in mind it can mutate. So therefore, we have taken the various epitopes. So if one epitope is really mutated, so we are not immune response on that epitope may not work, but other epitopes definitely will work. So that is what our strategy is.
Thank you, sir. So that was all. That's all the questions that we can take today.
Yeah.
Thank you again.
Yeah.
I would now like to request.
Yeah.
I would now like to request our convener, Professor Inare Banerjee, to give the vote of thanks to ma'am.
Thank you, Shinjini. Thank you, Professor Dash. Despite all the problems in between, I think it was quite worthwhile listening to you. You're a first-class immunologist yourself, and you've been working on tuberculosis and the lung for decades. So coming from you, I think this lecture would be very enriching for many of us. I was listening to the answers that you were giving to many of the questions, and I think the congregation probably has a lot more questions in mind. But as you said, it's a work in progress, and many of the things are still not known to us. So our best wishes for the work that you are doing in your lab, and we hope that we will get over this COVID menace soon and go back to our normal lives. So thank you very much to our speaker. I can see that you're probably having a power outage in your locality, and yet you have been very patient and you have enriched us with your talk. So Thank you very much for that.
And thanks
And
for
thank
having
you
me.
to
Thanks
participants.
thanks for giving me the platform for talking about some of the things what we are doing. Thanks.
Our pleasure.
Okay.
Our pleasure. So with that, I think we will end tonight. If anybody else from PPB has any announcement to make, but I would like to thank the people who are working behind the scenes to bring this to you. Shaunak, Sundaram, Dushant, who is controlling the YouTube live platform, Chinjini, who moderated this today, Nondita, who has also been working behind the scenes. You see, when you unmute yourselves and the speaker is saying something and the screen sharing gets all skewed, this is where these people who have been working behind the scenes come in, and a big thank you to all of them. And Shriya, who suggested the 3 dots, by the way, and got this going. And did I miss anyone? So that's the PPB team. So Shinjini, over to you, or Shaunak, if he has any other announcement to make. And a big thank you to everyone. Good night.
Thank you, ma'am. So that we'll be ending the session today. The details for the next session has been shared here, and you'll be getting the registration links soon enough. So with that, we are going to come to the end of the session. Thank you for joining us, and thank you.